Wiliam Huckle. I'm a member of the Biomedical Sciences Department in the College of Veterinary Medicine and have within the last year relocated my lab from the main veterinary college building out here to the Corporate Research Center in the ILSP, along with colleagues from vet medicine and biological sciences and human nutrition, foods and exercise. My research interests are in the realm of cardiovascular biology. We study growth factors that are produced in virtually any tissue and have the ability to promote the formation of new blood vessels in tissue that needs additional perfusion of blood. So this is a very interesting biological process and an essential process during development and during the normal function of adult organisms to repair and maintain blood vessels. But it's also an interesting process pathologically. As it turns out, there are a number of disease conditions that are characterized by excessive formation of blood vessels and the dysregulated action of some growth factors that are of interest to us. In particular, the growth of solid tumors is associated with the excessive formation of blood vessels. And there's been a great deal of interest and some clinical success in designing therapies that specifically target mechanisms controlling blood vessels in tumors. In terms of preparation for undergraduate research in my lab, what has worked well in the past, in my experience, have been students who have come in, if they are relatively junior in terms of their college experience, if they have come in with a considerable amount of advanced biology in high school or chemistry and so on. In high school, that tends to work out well. Or more senior undergraduates, third or fourth year students, have had more experience, not only classwork under their belt, but laboratory experience associated with various classes. That helps. Beyond that, motivation, as far as career ambitions, that it need not even be in science. There are a lot of interesting things to think about and research related to our work in the lab that doesn't even involve bench science. It involves public policy or public health issues. So we'll certainly be open to that sort of interest in a student. What I've done in my lab before has been to set up independent studies with undergraduates, which is something of a flexible process in terms of the number of hours involved, the number of hours of credit that would be carried with it with successful completion. So the number of credit hours would dictate the number of hours in the lab. Now, on a day-to-day basis, really what we do is study sort of the biochemical details of how this communication works. So we study how the cells that are receiving a signal maintain their state of responsiveness by producing receptor proteins on their surface that can recognize specifically these chemical signals from other cells. So one of the ways we study these proteins is to study the genetic mechanisms that dictate how much of them is made and what forms of them are made. We're particularly interested in multiple forms of the same protein, from a single gene product, how different forms of protein can be made that have rather different and in some cases opposing functions. So we study, getting down a little bit deeper in detail, study mechanisms of RNA metabolism, RNA processing, that are part of this choice between making one form of the protein or another. So there's a lot of molecular biology we do, studying the processing of RNA using cell systems where we make plasmids that encode different forms of RNA, and we are able to study in a variety of means the processing events that occur inside cells when this RNA is produced. So we do some basic molecular biology, cloning and expression studies, plasmid construction. I'm sitting next to a gel electrophoresis apparatus here that you all probably recognize. There's PCR we do. There's a PCR machine further back the bench. We have available to us also some nice protein analysis equipment. On the far side of the lab is a fluorescence imager that's very useful for studying proteins that have been fractionated in acrylamide gels. Look at post-translational modifications or relative abundance of proteins or other changes of interest.